I'm Jose Rodriguez, an assistant professor of chemistry and biochemistry at UCLA and a 2017 Beckman Young investigator. Dr. Arnold O. Beckman was a world-renowned scientist, businessman, and philanthropist. Dr. Beckman is well known for his inventions, including the pH meter. His instruments facilitate new discoveries in laboratories around the world. Dr. Beckman has inspired me as a scientist in a number of ways. I most relate to Dr. Beckman through our joint passion for science and unending curiosity. Dr. Beckman's phrase, "There is no substitute for excellence," is a special source of inspiration. My group searches for answers to scientific questions that address human health, benefit society, and provide us with new tools and knowledge to continue asking new questions. My work to understand the structural basis for biomolecular function requires the pursuit of excellence in scientific practice and in the training of other young scientists. The rewards for excellence are evident in our accomplishments. Our molecules pinpoint every atom in a complex structure and tell us about the interactions that give those molecules function. Many of Dr. Beckman's inventions were born from the need to improve how we measure chemical, biological, and health-related processes. The work of my lab is likewise driven by a similar need. Technological innovations have illuminated chemistry and biology for almost a century. From the first X-ray tubes to the modern electron microscope, technologies allow us to see the world in unprecedented detail. Our need for technical innovation continues to this day. The Arnold and Mabel Beckman Foundation allows my laboratory to continue innovating. To do so, we follow Dr. Beckman's rules for success and pose the pursuit of innovation as a solution to pressing scientific and biomedical problems. My group is interested in identifying the arrangement of atoms that give molecules their shapes. These include large and complex molecules, as well as small molecules with just a handful of atoms. Many of these molecules are able to form crystals, arrays of these individual molecules that can be visible to the naked eye. Invisible are the arrays of molecules themselves, lined up one next to the other. Crystals can form from molecules as simple as glucose to as complicated as a virus with tens of thousands of atoms. My group was funded by the Arnold and Mabel Beckman Foundation to pursue the structures of molecules that formed very small crystals, the types of crystals that you can only see under a light microscope or an electron microscope. Here you can see some of these crystals up against the E in a dime or the edge of that dime. We investigate these crystals by inserting them into a modern electron microscope, allowing us to visualize those crystals and collect diffraction information from the scattering of the electrons off of these crystals that then allows us to determine where exactly the positions of every atom are within these molecules. An example of a structure is shown here with every single atom in the molecule identified, labeled by our group, and then refined. I'm incredibly grateful to the Arnold and Mabel Beckman Foundation for allowing my laboratory to make new discoveries in atomic science and for helping us train the next generation of structural biologists.